All right. Let's see here. Welcome. So what we're going to do today is we'll have our first day of presentations. Let's see. So I've got, let's see, who have I got here? So I've got, looks like AISSA, Mehmet Ali, Muhammad, and Newman are on the docket for today. Again, maybe I'll go ahead and put the form. Let me see here. A link to the form that the rest of you should be using to assess the presentation. So, is there someone who wants to go first? Anyone?
Professor, I can start if you want.
I'm sorry, what was that?
I can start it. This is the AISSA.
Okay. All right. So AISSA. So what I'll do then? Let's see. Let me switch over to participants here. So I'm going to give you control over the meeting. Let's see. Make a presenter. All right. So now you should have control over. You can share your screen if you have slides or camera or whatever. Okay. So you can begin whenever you're ready.
Professor. Can you see my screen, Professor?
Yep, I can see it.
So if you want, I can start. Hello, everyone. My name is AISSA, HOUDJEDJ, and today I'm going to present one of our recent publication, which is titled Ranking Cancer Drivers via Betweenness-Based Outlier Detection and Random Walks. And the authors of this paper are Professor Jassim Artan. My name is also listed there, and Professor Hilal Kazam. And the authors are sorted by their last names. And this paper was published recently in BMC Bioinformatics. So this is the publication that I'm talking about. So first of all, I will start talking, and I will give a small introduction, talking about some biological background and some computational background. First of all, I will start talking about biological background. As I mentioned in the title, it's about cancer driver genes. So this publication is related to cancer area. So I will be I will define briefly the cancer in few words. A cancer is a disease of genes which causes cells to grow and develop out of control. So a cancer develops when basically the body's normal control mechanism stops working. And here, the structure of cancerous cells will look very different to normal cells. And the problem here is that all cells do not die, but instead it will grow out of control, forming new abnormal cells. And we do have the hundreds different types of cancers, and according to the World Health Organization, globally, 1 in 6 deaths are due to the cancer. So this is briefly the definition of cancer. So it's the growth of cells out of control or develop out of control. It's called cancer. So somatic mutation is also another biological terminology. So somatic mutation is an alteration or a change in DNA that occurs after conception. And somatic mutations can occur in any of the body cells, except the germ cells. So we cannot find the somatic mutations in sperm and egg, which means that this somatic mutation or it cannot be passed to the next generation. And the main point of somatic mutations here is that this mutation can be the cause of cancer development. And now I will be briefly describing and talking about some type of genome variation, starting with SNV. SNV is single nucleotide variation. So SNV mutation is the mutation substation of a single base in the DNA sequence. And also it can be, so this SNV can be common for a population and non-common for another population. And if the variant is present in at least 1% of our population, then we call it SMP or single nucleotide polymorphism, as I mentioned here in this picture. And another type of genome variation is in this. In this stands for insertion and deletion. So in this is the short of in this are the insertion or deletion of a small segment of bases from the genome. As you can see here, let's say that so this is our original sequence. Okay, if you do have a deletion, so one single base here will be removed from our original sequence. So all the other remaining sequences will be shifted to the left. If you do have an insertion here compared to the original sequence, then all the remaining sequences will be shifted to the right side. So in this, so this stands for insertion and deletion of one genome base here. And finally, CNV, which is called copy number variation, is a concept where a region of DNA is either duplicated or removed. So instead of compared to SNV, in this here, we do have only one single base here to be deleted or inserted, but in CNV, we will have a sequence of genome bases will be duplicated or removed from our original sequence. So just to move forward, I'm not going to focus that much in this genome variation. But I'm just giving you an idea of how these alterations are happening. So I'm not going to talk too much about this biological background in the rest of my presentation, but just to have an idea about how these alterations are happening. So I'm just giving this small introduction. Now, I will be defining new terminologies in bioinformatics, which are driver mutations and passenger mutations. So very quick, a driver mutation is a mutated gene that's mostly selected in the tissue where the cancer arrives. So this driver, this gene, the gene that is found in a cancerous cell, mostly are called driver mutations. And these genes are given cells a growth advantage when they are mutated. So that's why we call them driver genes. And the other terminology, which is passenger mutation, are these somatic mutations that have no contribution to cancer development, but you can't find them in a cancerous cell. So the main point here is that if you can identify these genes, then this is a really important step towards personalizing treatment and finding some therapies for cancer. If you can distinguish between this driver mutation and passenger mutation, then we are given a really, so we are showing really good steps toward finding a therapy for cancer. Now, so I'm done with biological background, but now I will be talking about some, let's say, computational background. So starting with protein-protein interaction or PPI. So PPI is a network that gives us the chance for a better understanding of interaction and connection between proteins and genes. So the understanding of protein interaction and gene interaction between them it leads us and push us to develop drugs and therapies as well. So these PPIs are constructed using some computational techniques and many physiochemical techniques as well. And nowadays, a few proteins are discovered. I think less than 20% of the protein are discovered yet. So that's why there is a huge need for new techniques to predict non-discovered ones. So this is about PPI networks. And also biological pathways. As you know that most of the molecules in a set are proteins and small molecule compounds. And these compounds are working and interacting together in order to perform some tasks and accomplish a mission in a cell. So in few words, a set of molecular events that end in the creation of new molecular component or change in cellular state is called biological pathway. And if there is any perturbation to these biological pathways, if it's not performing, for example, properly, or there is some alteration, it could lead to several diseases such as, for example, cancer rise or diabetes and many other diseases. So these biological pathways are the sequence of events that some proteins are performing together in order to accomplish a mission. So there are multiple databases and projects that provide such information about biological pathways. In this publication, we are using two databases, KEGG and Reactome. And these databases are designed to for us, for researchers and for bioinformaticians, to validate and interpret the results of experimental studies and research. It's also used to validate the results of bioinformatics algorithms. And also, both this KEGG and Reactome are open source databases. So it could be used freely to validate algorithms results and many other experiments. Another terminology or another database here is gene ontology or Go ontology. So Go is a project that aims to map genes and their products to biological annotation. How can I say, maybe, so it's a Go term or annotation describes how a gene is related to a molecular function. So gene ontology is used to describe the functionality of genes. So it's basically a graph where we have nodes as gene or proteins, and the interaction between these nodes are describing the function or the relationship and functionalities that both genes are sharing together. So this is gene ontology. So it's used for functionality analysis of genes and proteins, and also it's used to validate the results of algorithms as well. Yeah. So now, after finishing talking about some biological and computational background, now the main question is that why we work on such, or why we try to write such a publication, or why we started working on such a project, and what is the aim, and what is the main goal. So as I mentioned before, that the cancer is developed and caused by somatic mutations. Now, after describing what or defining what somatic mutations, now we can call it as driver genes or set of driver genes. So the goal is that are we able to develop a new method that, or a computational method that identifies the cancer genes among the somatic mutations that we have? Are we able to distinguish, or are we able to distinguish driver genes from the passenger mutated genes? And also, can we provide a better method to compare it to the previous state-of-art methods that are used or implemented in order to prioritize driver genes? If yes, then you can help in the development of treatment and therapies for cancers. So this is the main question that we started with, and this is how we defined our challenge and our hypothesis. So our method is called betweenness, or defined why we call it betweenness. It's compared against 5 other state-of-art methods: betweenness, DriverNet, DawnRank, and Subdyquency, and finally into drivers. So these 5 methods are methods that use the same input as our method, and at the end they output a set of genes that they prioritize, or they claim that these genes are driver genes for such a cancer type. Okay, so I will be talking about this method one by one. So first of all, I will start talking about DriverNet. So DriverNet is one of the first methods that implemented bipartite graph, and it was published back in 2012. Yeah. And so for those who are not familiar with bipartite graph, so bipartite graph is a graph with 2 partitions. As you can see here in this picture, I do have a graph with 2 partitions, left one and the right partition. So the idea of bipartite graph is that you can have some connections or edges between nodes from different partitions, but you cannot have edges between the nodes of the same partition. So this is, this is the general idea of bipartite graph. So here, what they do as an input for their method, they do have PPI network. Okay, the PPI network is protein-protein interaction, which is a matrix where we have genes as rows and genes also as columns, and it's a binary matrix. One, if there is a connection in the PPI network between that gene and the other gene, otherwise it's 0. Gene expression is a matrix where we have patients as columns, for example, and genes as rows, and the entries of this matrix is the gene expression values. It's how that gene is expressed in that cell or that given patient, for example, or sample. A mutation matrix is also a binary matrix, so we have patients as columns and genes as rows, and it's a binary matrix, one if that gene is mutated in that patient or not. So this is the input for the method. So what they do, let's say that they have, for example, they're focusing on breast cancer, okay, and they have 500 patients, and what they do here is that they took all the union of the mutations across all samples, and they form this left partition of our bipartite graph. So here on the left partition, we'll have a set of all mutations across all patients that we have in our data set. And for the right partition here, they select for each patient, as you can see here, they do have multiple patients here, and for each patient, they do have a set of outlier genes. Now, how they define these outlier genes is that, for example, let's say that they are focusing on this gene. Okay, this is PIK3R1. This is a gene, and as I mentioned, that for each gene, we do have gene expression values across all samples. They plot the distribution of the gene expression values across all samples, and they found that it fits a normal distribution, as you can see here. Right? As you can see here, and based on this distribution, they set a threshold for this gene, which is equal to 2 multiplied by standard deviation plus the mean. So now for each patient, they check the gene expression value. If it's 2 multiplied by standard deviation plus the mean value, it's greater than 2 standard deviation plus the mean. If yes, then they consider the gene as an outlier for the patient. After that, now for each patient, they will have a set of outliers, so it could be different from patient to patient based on the gene expression values and mutations on the left partition. And the edge between the mutated gene and an outlier gene is defined if there is an edge between these 2 genes in the PPI network. Okay. And for the final ranking, what they do here is that they rank mutated genes, because we are focusing only on the mutated genes. They rank these mutated genes based on their degree. So they select the gene or the mutated gene with the highest degree, then they select it as our first, for example, driver genes, and they remove these mutated genes and all outliers connected to it, and they recalculate the degrees again, and they select the next highest mutated genes with the next mutated gene with the highest degree, and so on until they cover all these outlier genes. So this is the final ranking of this method, DriverNet, which is published back in 2012, which is one of the best methods and one of the first methods that they implemented by partite graph in their method, and many other methods they came after this method. They use the same concept by making some small change to their methods. So another method which is Subdyquency, which is published in 2020, I mean 1 year ago. So the idea of Subdyquency method is formulated in the same way as DriverNet. They do have bipartite graph, they do have mutated genes on the left partition and outlier genes on the right partition. But compared to DriverNet, it was an unweighted graph, so we don't have any weight between the driver genes, between this, for each edge between this mutated gene and outlier gene, we don't have any edge weight. So Subdyquency, they come up with a new method. It's not a new method, but a new thing on another value to DriverNet method, which is adding some weight, and also they try to construct random work on this bipartite graph. So after they create a bipartite graph, as I mentioned before in DriverNet, they run a random work on the bipartite graph. Okay, if they run random work on the bipartite graph at the end, so here they will get a matrix like this one. They do have patients as in columns and mutated genes as rows, and for each mutated gene they will have a score. Okay, and the sum of these scores across all patients is defining our final score for this mutated gene, and ranking them in a non-increasing order is defining our final ranking list. So the ranking list is used also as our final ranking. So this ranking is defined as our driver genes outputs from Subdyquency method. Another method, which is DawnRank. DawnRank is another method that we compared our method against, which is published back in 2014, where they use the same input data compared to Subdyquency and DriverNet. And this method is implementing Google's PageRank algorithm with small changes in the core code. And the idea of the algorithm is based on the hypothesis that if a mutated gene, let's say for example M, has a large direct or indirect connectivity to a differentially expressed gene. So differentially expressed genes is the outliers genes, okay? Is the set of outlier genes that we defined before. So these ones are called differentially expressed genes or dysregulated genes. So if mutated gene M, for example, has a large direct or indirect connectivity to dysregulated gene, then the gene has a higher impact score. That's why they use Google's algorithm to do the ranking for the ranking. So for the ranking of each gene is defined iteratively as follows. R T+1 is the rank of in the fifth iteration, which is equal to 1 minus D. D is a value between 0 and 1. It's a constant. Okay, it's 0.5 here, multiplied by F. F is the gene expression matrix. Okay, where we have patients as columns and genes as rows, and gene expression values as entries, plus D. D is the constant between 0 and 1, multiplied by M. M is the matrix, as you can see here, which is the adjacency matrix or the PPI matrix, where we have genes versus genes. 1 here, if there is a connectivity in our PPI between this gene and this gene, divided by the degree is used for the normalization. Okay, and this one is done iteratively until it converges, and at the end we will have mutated genes, and we do have scores. The sum of scores is denoting our final ranking for DawnRank method. So this is about the state-of-art method that we are comparing our method against. So now I start talking about our method and what's the difference between our method and the other methods, and what's the added values of our methods, and what's the how we can say that our method is better than the other methods, and the evaluations and everything. So as an input data here, first of all, the other methods, what they do is that they use the gene expression values from only the tumor cells for that patient. Let's say that for patient A, I may have gene expression values from a tumor cell, and also I may have gene expression from a healthy cell or normal cell. Let's call it normal cell. So here, what we said is that we constructed our input data to be a data set of paired samples. So for each sample, we should have gene expression for a normal and also for a tumor. So we are including only samples with both information from normal and tumor. Among all cancer types that we have in TCGA database, what we do is that we set a threshold to set a cancer type to be added to our evaluation. So the threshold is 10. So for each cancer type, breast cancer, lung cancer, head and neck cancer, for example, we check if you have at least 10 patients with both normal and tumor samples. If yes, then we use that cancer type in our evaluation. Otherwise, we don't include that cancer type, because at the end we are constructing our pan-cancer data set, which is the union of all cancer types that provides at least 10 samples with both normal and tumor gene expression data. So as I can show you here that, first of all, I started working with breast cancer. Then at the end, I come up with results for 14 different cancer types here. So for breast cancer, I do have 112 patients, and for lung, I have 16, for example, for LEST, which is lung, and LOAD is also lung, which for 45 patients. And the union of all these samples to define our pan-cancer data set is denoting our final size of samples, which is 580 samples here. So in our publication, we provide an evaluation on breast cancer and pan-cancer, and also lung. Lung is the union of CSE and LOAD here. So after that, I constructed the PPI network by making some pre-processing on the IntAct PPI network that was downloaded from IntAct database. At the end, they come up with a PPI network of 15,345 nodes or genes or proteins, and 113,524 edges or connections between these genes. So this is a PPI network that I'm using. And as you saw in the title, there is betweenness-based outliers. So I will here define what is betweenness. First of all, betweenness is a measure of centrality in the graph based on shortest path. So in which it gives us an idea about how a node is really important in a graph. So let's say that this is my graph here, okay? And for every pair of graph, I must have at least one shortest path between two nodes. Let's say that I want to calculate the betweenness. So we need, let's say that for selected node, if I want to calculate the betweenness value of that node, first of all, I need to go through all the pair of remaining nodes, excluding that node. Let's say that if I want to calculate the betweenness value of the node 4 here, I need to get all the combination of pairs of the remaining nodes, excluding this node 4, okay? And for each pair of vertices or nodes, we count the number of shortest paths between them, which is denoted by sigma st, which is the denominator here, which is the number of the shortest path between two pair of vertices. Let's say that I'm focused on a node 4, and I will take the node 6 and 0. So how many short paths I have? I have 2 short paths. I colored them by red here. 6 to 4, 4 to 2, 2 to 0, short path of size 3, of length 3, and the other one is 6 to 5, 5 to 1, and 1 to 0, which is another short path of size 3, sorry, as well. So I do have 2 shortest paths, okay? And how many short paths are passing by the node 4, which is our focusing node? I have only 1 short path here. So for the pace 6, 0, the value will be 1 divided by 2. The number of short paths that passes by the focusing nodes divided by the total number of short paths. So here at the end, I will get the sum up of all the pace values here. At the end, it will be not the final betweenness value of the node 4 here. So the higher this value is, the more important this node is in our graph is. So because if most of the shortest paths are passing by our focusing node, which means that it's really centralized in our graph. So I skipped the method betweenness before, just to include it here after determining what betweenness is. So this method was used by Professor Jatin and DePazzo in one of their publications in 2017, in which they compared the graph theoretical measurement to prioritize driver genes. So as input, they use PPI as I mentioned before, and somatic mutation data, which is a metric that denotes patients as columns and genes as rows, one if that gene is mutated in that patient or not, and gene expression data of paired samples. So they include only samples with both normal and tumor gene expression data set. The same as what we are doing. So here, for each sample, they constructed 2 graphs. 2 graphs, a normal graph and tumor graph. Okay, for each patient, they construct 2 graphs, normal graph and tumor graph. First of all, both graphs are the same, which is the PPI network, which is the same as PPI network. Then for the normal graph, they remove lowly expressed genes by checking the value of the gene expression in if it's less than 1. So they set a threshold for expressed genes. If it's expressed genes, it must be greater than 1, otherwise it must be removed from normal graph. And also for tumor graph, they remove also this lowly expressed genes. But for tumor graph, they also remove mutated genes. So for normal graph, they keep mutated genes, but for tumor graph, they don't. So they remove these mutated genes. And they calculate betweenness values for each graph. Then at the end, we will have 2 matrices for 1 patient. So we will have betweenness value for each gene in the tumor graph, and also betweenness values for each gene in the normal graph for each patient. And to do the ranking, what they do is that they sum up the absolute betweenness difference values between normal and tumor graph across all patients. And at the end, we will have a score for each gene, and sorting down these values in non-inclusion order is denoting our final ranking. So this is the final ranking. So this is, I'm talking about the method betweenness, which is published back in 2017 by Professor DePazzo. And we just try to use the same idea here. So as I mentioned before, I have data for normal and tumor. For each patient, I have data from normal and tumor tissue. So at the beginning, I will have the same graph, okay, which is the PPI network. And for the normal graph, I'm going only to remove genes with less lowly expressed genes. I'm not going to include, let's say this gene is lowly expressed in patient I. So I'm going to remove it, and also this gene and this gene. So this is the normal graph. And for tumor graph, if this gene is lowly expressed, I will remove it, and this one, if it's lowly expressed, I will remove it as well. And also I'm removing mutated genes. I'm not going to include them. Why? Because at the end, so mutated genes are included here and are not included here. So when we calculate the betweenness values, so the betweenness values for mutated genes in the tumor graph will be 0 because I'm not including them, so the betweenness values will be 0. But the betweenness value of mutated gene here will be some value greater than 0. Maybe, I don't know, thousands or 5,000, depends on. And when I calculate the betweenness difference for that mutated genes between the normal and tumor, so I'm going to assign high score to that mutated gene, which that going to rank them in the top because of this betweenness, the absolute betweenness difference. So so this is the same idea as I'm talking about betweenness calculation. Now, very different from DriverNet, Subdyquency, and DawnRank and the other methods, in which they select outliers based on the gene expression distribution. But here we set we using the absolute betweenness difference values for that gene instead. So for each gene which exists in both normal and tumor network, we plot the betweenness difference values across all the patients. So here is, instead of plotting gene expression, I'm plotting the betweenness difference of this gene across all samples. As you can see that the distribution are not hitting a normal distribution. It's only of one tail, which is called truncated truncated distribution. That's why I'm using a library which is called TrankNorm, which is used to estimate the mean and standard deviation. So instead of plotting the gene expression values and select the outliers, now we are plotting the absolute betweenness difference between normal and tumor tissue for each sample, and select our outlier. So this is our this is an added value to the previous method. At the end, we are going to construct a bipartite graph. Mutations on the top and outliers based on the betweenness difference are on the bottom here. Okay, and the edges between mutated gene and outliers are based on PPI network. Then I'm going to run a random walk on the bipartite graph. So at the end, when running the random walk on our bipartite graph, I will come up with the matrix like this one here, genes on the M by M matrix. So here the entries are the random walk. So it's the random walk score for that gene after I iterations, for example. So this is the probability of or the output of the final distribution of our work after I iteration. So now to rank our mutated genes, we assign a score for each mutated gene, which is equal to alpha. Alpha is the value between 0 and 1. And in our method, we selected alpha equal to 0.5, which is equal to the degree of mutated gene I divided by the maximum mutated degree of mutations, just to denote the normalization here, plus 1 minus alpha multiplied by the sum of incoming edge weights for gene I after random walk. So here I'm going to sum up all these incoming edges scores after the distribution of the work, and I will divide it by the sum of the maximum value of it, just to denote just for the normalization here. So this is my final scoring for each mutated gene. I will do the same thing. Then at the end, I'm going to sort them in a non-increasing order, and this is my final ranking of my method. So just to summarize the methods, first of all, I have PPI network that I got it from the Interact database, and I have paired normal and tumor gene expression data and somatic mutation data that we got it from TCGA database. And I constructed 2 graphs, a normal graph and tumor graph. For normal graph, I'm removing only non-expressed genes, but for tumor graph, I'm removing normal genes, non-expressed genes, sorry, and mutated genes. And I'm going to calculate betweenness centrality on normal graph and tumor graph. At the end, I will have, after running betweenness centrality, I will have two matrices. One is for normal graphs and the other one is for tumor. For each gene, I will have the betweenness value in patient, for example, patient 1 until n, and do this. I will have the same thing for normal. And at the end, I'm going to calculate the absolute betweenness difference between tumor and normal. I will have this matrix here, which is then I will generate or construct the outliers based on this, based on the betweenness difference values. Then I will construct my bipartite graph, run the random walk. I will get this scores, which is random walk step scores of probabilities, then calculating combined score, which is alpha multiplied by the degree of mutated genes, plus 1 minus alpha, the sum of the scores. Here, at the end, I will have, for each mutated gene, I will have a score, ranking them in an increasing order is my final ranking. So this is my method. Which is called betweenness because I'm using, so net is for network and between is from betweenness. And for the result and discussion here, for the validation part, first we compare the method based on their ability to recover the set of non-cancer genes. So we do have some reference sets or some cancer sets, for example, CGC, CancerMine, NCG. So these are a set of non-cancer genes. So we know that these genes are contributing to cancer arise. So CGC, for example, is the world's largest and most comprehensive resource for exploring the impact of somatic mutations in human cancer. And also I have NCG here, which is the network of cancer genes, which is manually created for 2,372 genes, I think, whose somatic modifications have known or predicted as cancer driver genes. So basically what they do here, they come up with a set of papers in which they cite some genes as, or they predicted some genes as cancer driver genes. That's why, so the union of all these genes, they use the union of these genes to construct some reference sets, for example, CGC and CancerMine. CancerMine is also a literature-minded database of drivers in which they, for CancerMine, the data set is presented as following: the gene names and the cancer type and the number of citations. So for each gene, we do have its cancer type, which is lung cancer, breast cancer, or any other cancer, and also how many times this gene is cited as a cancer gene. So that's why I come up with CancerMine 3 and CancerMine 5, which means that CancerMine 3 are the genes for lung cancer that has been cited at least 3 times. CancerMine 5 at the cancer genes for lung cancer, for example, that has been or have been cited at least 5 times. So this is our reference set that we are going to compare our ranked genes with. So this is our reference set. So we first compare the method based on their ability to recover the set of non-cancer genes by calculating true positive traits and false positive traits. And for each method, DawnRank, Subdyquency, DriverNet, and our method, what we do, we are taking the top 1,000 genes and calculate TP and TPR and SPR values. As you can see here, so betweenness, so this is the fraction of recovered reference genes for lung cancer. I'm focusing on lung cancer here. You do have CGC cancer genes as reference set, CGC rare, and CancerMine 3. So here, this is the true positive rate and false positive rate for doing it for top 1,000 genes. And as you can see that betweenness is achieving higher area under the curve than all the alternatives for CGC and CancerMine 3 here. So betweenness is beating all other methods for CGC and custom 3. But for CGC array, it has the same area under the rock or the curve for our method and DawnRank, but it's better than the other competitors. So, which means that our method is showing really good performance comparing to the other methods for the lung cancer. And for breast cancer, I did the same evaluation here. As you can see that DriverNet is achieving the top performance with CGC and CGC array reference set. But for custom 3 here, I do have a problem with DawnRank. So DawnRank method is performing even better, slightly better than our method. But it's still we are still doing good in 2 over 3 of our evaluations here. So this is for breast cancer that I said, and this one is for pan cancer. For pan cancer, I'm taking the sample of patients across all cancer types that we have. As you can see that for CGC, our method is not performing that much good compared to DawnRank and compared to Subdyquency. So this is Subdyquency is doing pretty much good compared to our method. And for CGC array, our method and the first competitive best method and our method are performing the same according to the area under the curve, as you can see here. And for custom 3, our method is the second best method compared to Subdyquency with slight difference. So 0.0166 compared to 0.0167 in terms of false positive rate and true positive rate. So I know that this kind of topics maybe it's boring for non-bioinformatic for non-bioinformatician researchers. So that's why I tried to skip some steps and some biological information in order just to keep it as computational method and not going into biology that much deeper. So as a conclusion, so as I mentioned, that implementing DriverNet to identify outlier genes based on DriverNet neutrality is one of the main contributions, which shows a good performance of our method. And also the usage of random walk have added values in the identification of cancer driver genes. So in the paper, I added a section in which we investigated the top ranked genes. We took the top 30 genes that they don't show up in the reference set in CGC or custom CGC array or NCG, where they have a role in cancer development based on iterators. So we discovered some novel driver genes as well. You can see them in the paper. I listed for each cancer type, I listed the top 30 genes that they don't appear in the reference set. So as a future work, so the problem is that our approach or our method compared to the other methods is based on paired data sets, in which it makes it critical and quite limited because we have small cohort sizes, or we do have small number of patients with both normal and tumor. For example, for breast cancer, we do have 900 patients or 900 patients with tumor data set, but only 112 patients with both normal and tumor. So it's almost 1 over 9. So it's really critical and quite limited. That's why it pushed us forward to think more about providing or constructing some computational algorithms or methods that predict and generate normal samples for the missing ones. And also here we are using bulk expression data sets in which even if it's from tumor cells, okay, the data is from tumor cell, but it may contain some non-cancerous cells as well. So non-cancerous cell can be also present in the data, which is a little bit noisy. So many research and publications nowadays are suggesting switching to single cell, which is they select only the cancerous cell instead of bulk data. As I mentioned, that our method is showing some low performance for pan cancer. That's why maybe we are thinking about introducing some more information in our scoring function that it may improve our results. Yeah, that's it, and thank you so much for your listening. I hope that it was a good presentation for you. Thank you so much.
All right, thank you, AISSA. Okay, so does anyone have any questions? I mean, let's see, we have about an hour and a half left. Got questions? Someone?
Teacher, I have some questions.
Sure.
I'm not so close to bioinformatics, but I'm interested in the computational side of this work. It's not clear to me what's the time complexity of this work.
Time complexity of?
Of this work. For example, your methods source these cancer-driven genes faster according to others.
Okay, yeah. So as I mentioned, we are introducing something new here, which is the betweenness calculation, which is something different to the other methods. So for betweenness calculation, it takes some time because imagine for each patient will have two different graphs, and so for pan cancer I will have 1,000 graphs, I think. Yeah, I will have 1,000 graphs. So it takes some time compared to the other methods. And also for pan cancer, I have huge matrices, which is about 500,000 by 500,000. So it takes some time compared to the other methods, but for the running time. But in terms of results, it shows that good performance compared to the others. For pan cancer, it takes some time, but for lung cancer and breast cancer, or small cohort, it's not. I mean, it's the same. You can say it's totally the same.
Okay. Maybe second question is related about the if this numbers of the samples increases, then
Yes.
still this achievement is still stable.
Yeah. So because as I mentioned before, I started with lung cancer. In lung cancer, I have only 41 samples or 61 samples, and I tried to increase it to breast cancer, and I have now 112 patients. And also every time I try to include a new cancer type just to increase the cohort size, and it shows good performance until I come up with this pan cancer data set, which contains a larger number of 508 or 580 samples. So every time when we increase the number of samples, so we are including more mutations here. And whenever we include more mutations here, we will have more outliers as well. And also, we will have more connections between these mutations and these outliers. And when increasing these mutations and the size of mutations and the size of outlier set, so we are given more chance for the degree parts of our scoring. Let me just say. Yeah, so we are given more chance for this degree part for the driver genes to score at the top. Yeah, so this is our hypothesis.
Okay, thanks for answering.
You are most welcome.
I have a question.
Sure.
Okay. How many times did you train the model from scratch using new random weights initializations?
Sorry, how many times?
Yes.
Can you say it again? I didn't hear you.
Okay. How many times did you like restart the training from scratch with new weights, reinitializing all the weights of the neural network part? How many times did you do that?
How many times I started from scratch too? I cannot hear you clearly. I don't know if it's only for me or.
Maybe I'll just type. I can type, and then you can. Is that better?
Okay, sure.
Yeah.
Do you mean neural network? Because I'm not using any neural networks here.
Right. So the neural network, when you initialize it the first time, you're supposed to initialize it with random values for the weights, and so.
Okay. So I'm not okay. I'm not initializing it with random values. At the beginning, I'm initializing the values with mutation frequency. Okay. For each mutated gene, for each patient, I'm calculating how many times that gene is set as mutated genes across all samples divided by the number of cohort types. So my first initialization is the mutation frequency.
That's for the weights of the neural network?
Yeah, this is for the random walk factor.
Okay.
Yeah, I don't know if it's Aisa, are you talking right now? Because I can't hear you.
No, I'm not talking. I'm just waiting if there is any other questions.
Okay. Yeah, sorry, my network.
Okay.
You said for some reason so anyway, I moved closer to the router and I'm hoping that may work better, but I just wanted to make sure. All right, so if that's it, if that concludes that, let's go ahead and set up for the next presentation. If someone else is volunteering, we can do that. I think we can probably, if everyone else can kind of stay around 30 minutes, and that would include question and answer time as well at the end. Then we can get 3 more in today. So let's try to do that. So is someone else going to volunteer?